Hello, my name is Mohamed Noor. I'm a biology professor at Duke University, and I'm an occasional science consultant for the Star Trek universe. I'm so excited today to talk with you about biology and science consulting in Star Trek Discovery. This will be a full-length convention talk that I will have actually given at some conventions too. So I hope you enjoy this online, and thank you for joining us. Let's dive right in. So I'm going to start first with an outline for today's talk. I'm going to first talk about some science tidbits from seasons 1 and season 2 of Discovery. Now, this will also include a little bit from Short Treks. I should stress, I did not consult on either of these two seasons, but I just picked some science bits that I thought were really cool, and I hope you enjoy them as well. Then I'll talk about some science that I actually worked on for seasons 3 and season 4 for Star Trek Discovery. Some of this was collaborative, a lot of it with the science advisor for the Star Trek franchise, the amazing Dr. Aaron McDonald, but also some pieces that were collaborative with some other folks as well. And then I'll close out the talk talking a little bit more generally about what it's like being an occasional science consultant for Star Trek. Now, I want to stress one thing, which is very important. I am a biologist, so I'm only going to be talking about biology in these depictions. I'm not going to be talking about physics, so I can't explain the warp drive or transporters or any of those things to you. I apologize. That will be somebody else's talk, I hope. Anyway, without further ado, let's dive right in. For season 1, I picked 2 interesting biology topics that are very short to cover that I just want to comment on briefly, and I picked 2 that I want to go into in a little bit more depth. I bet you can guess one of those. Anyway, let's start first with one of the short ones. There was a quick comment in the very first episode, the pilot episode of the series Star Trek Discovery called The Vulcan Hello, about DNA unspooling. You might remember Michael Burnham was sitting in the Shenzo's medical bay, and the doctor said, "Wait, the recombination process is nowhere near finished. Do you understand the effect of genetic unspooling? You don't want to die that way." Very dramatic. I was very excited about this because when I heard the mention of recombination, that's something that my research laboratory actually works on. So I was like, ooh, recombination, that's great. And the unspooling thing is especially cool. So let's talk about what was happening there and what this doctor was actually referring to. You might remember shortly before Michael Burnham ended up in the med bay, she was flying like an astronaut just in space there, headed towards the Klingon ship. As this was happening, she was being exposed to galactic cosmic rays, a whole bunch of other radiation too. Radiation is really bad for you in a lot of ways, but it's also bad for your DNA in a couple of different ways. Now, when we think about DNA, we tend to think of DNA as this long thread-like thing, which is kind of true. But in fact, it's not hanging out in your cells as this long thread, just loose like that, but it's actually tightly wound around these proteins that are called histones. Now, recombination is a process that repairs broken DNA by using the unbroken copy, because you have 2 copies of your DNA. It uses the unbroken copy as a template. Now, for the recombination to happen, you have to unspool it from these little histone proteins. So unspooling happens during repair, but you can't stay unspooled without the DNA being at risk. So what I infer is going on here is that either the DNA was very severely damaged, which was, affecting how it would spool, or the histones themselves were broken. Either way, this was some pretty cool science that was just slipped into a passing comment in Star Trek Discovery's pilot. Just a couple of episodes later, in episode 3, we saw a reference to some microbes that can get energy from electricity. This was while Michael Burnham was on a shuttle. This is before she actually ended up on the USS Discovery. They referenced species GS54, which feeds on electricity. So this got me to look up, are there any microbes that can actually get energy from electricity here in the real world that we know about? And in fact, there are some Earth microbes that can directly accept and use electrons from electrodes for energy. These are in the genus called Geobacter, and they specializing in making electrical contacts with electron acceptors. So this is really cool. This is actually a real thing. There's multiple scientific papers about it, and yes, this is not at least a crazy concept. Now, let's go into 2 more things in a little bit more depth. Those were just some little appetizers, but now let's get into the meal here. A big one that we saw in Star Trek Discovery Season 1 was the tardigrade, also known as Ripper. You may be familiar with tardigrades. They're often called water bears. They're found just like on little bits of moss, maybe out in your yard, things like that. They mostly eat algae. There's a few that are carnivores, and some will eat smaller tardigrades too. Now, Ripper, the one that we saw in Star Trek Discovery, does kind of resemble some Earth tardigrades in some ways, but he's really different in other ways. So like the Earth tardigrade, they have the same kind of overall appearance, a little bit like a roly-poly sort of. Like Earth tardigrades, we saw the Ripper can dehydrate and can live for years all dried up. This is also true for Earth tardigrades. They can actually dry up and live for years dried up as well. Now, there's a lot in the popular media about how tardigrades can survive the vacuum and radiation of lower Earth orbit. I'm going to come back to that in just a second, but that again at least matches what we hear about tardigrades all the time. I'm going to come back to that though. Now, some differences between Earth tardigrades and Ripper were, first of all, Earth tardigrades don't have antennae, right? They don't have an internal skeleton. We actually saw a scan there of Ripper showing a skeleton inside. Earth tardigrades don't have that. Earth tardigrades don't have jointed legs. They just kind of have those little stubs. Earth tardigrades are aquatic, meaning they don't actually have lungs. You might remember Ripper actually could scream. But the biggest difference, of course, is size. Ripper was gigantic. Earth tardigrades are microscopic. Now, there's a common misconception in a lot of sci-fi that you can take something that's tiny and just make it really big. That actually doesn't work. And the reason that doesn't work is for something called the square cube law. So volume grows faster than area. For something like a cube, volume is length times width times height, whereas for something like a square, area is just length times width. Now, the cross-section of a muscle will grow much slower than the mass they're supporting, because the cross-section of the muscle is using surface area, whereas the mass is growing as the cube. So let's say, for example, you're going multiples of 2. You go from 2 to 4 for surface area. You go 2 to 8 for volume. Now, as you do this more and more and more, you eventually have something that's so big that the muscles can't support it. This is why small animals like ants seem much stronger than they really are. But if you made an ant gigantic, it actually wouldn't be that strong at all. It would fall apart under its own weight. So we have to assume that Ripper is dramatically stronger than Earth tardigrades, given its dramatic increase in size. Now, let's talk for just a minute about this surviving in space thing about Earth tardigrades. There's lots of hype about this and their ability to survive in space. You might remember there was a story recently about some tardigrades that fell to the moon and maybe they were still alive. Yeah, they're not alive. But here's a headline from Popular Mechanics: 7 fascinating facts about the tardigrade, the only animal that can survive in space. Now, I want to show you the actual scientific study that looked at how well tardigrades survive in space, where the European Space Agency took some up there and actually exposed them to the vacuum of space and radiation and things like that. So in this figure that you see here, the rows are 2 different tardigrade species they studied, and the columns show survival of adult tardigrades over time and hatching of eggs. So first of all, let me just say, none of the eggs survived when exposed to both vacuum and all forms of UV radiation that you see in space. 0. When you look at survival of the adults, one of the tardigrades also had 0 survive at all. Like, none of them. The other one had this, you see the little circle I have there on the bottom? They had a tiny little number, I think it was literally 3 out of around 100, survived for a couple of days after having full space exposure for a couple of days. So by and large, I think this is a little bit overstated in that, yeah, I can survive in space, a tiny bit of them, of one species, for a very short length of time. But, still really cool. I mean, that's still better than I could do or better than humans could do. So fair enough. Now, one other thing that came up about tardigrades in the series was this reference to horizontal gene transfer. Okay? I'm going to play a little clip.
Like its microscopic cousins on Earth, the tardigrade is able to incorporate foreign DNA into its own genome via horizontal gene transfer.
This was actually really cool that they incorporated into Star Trek Discovery, because in 2015, just a little bit before Star Trek Discovery premiered, there was a paper published in the Proceedings of the National Academy of Sciences, which is a really reputable journal. This was the first full genome sequence of a tardigrade, and they found, and it's shown here in the title, evidence for extensive horizontal gene transfer. So that's really cool. This actually got a lot of publicity. My good friend, Dr. Aaron McDonald, always likes to point out the first author of this paper was Boothby. Just a coincidence. Anyway, this got a lot of press, and that's probably why they went ahead and incorporated that into the series. And in fact, the authors argued that this horizontal gene transfer may be what facilitates its broad tolerance, including in things like space. Unfortunately, the paper was just wrong. They thought they had it right, but it was just wrong. And in fact, the next year, there were many papers published showing that, in fact, there's not extensive horizontal gene transfer into tardigrades. That said, unsurprisingly, the original result got a lot of publicity. The papers showing it was not correct did not get as much. Still, I applaud the showrunners and the writers for trying to incorporate some super recent science into the series. So I still call this a win nonetheless. Now, last but not least, let's talk briefly about one of the highlights of Star Trek Discovery, which is the spore drive. So this is one that takes a little bit of extra explanation. Now, interestingly, you can get a lot of this explanation, not necessarily just from me, but in fact from somebody whose name is Paul Stamets. And this is the real world biologist whose name is Paul Stamets, who actually is a mycologist, meaning he actually studies fungi. Now, he has a fantastic TED Talk that you can see. I'm going to play a little clip here.
This was first discovered in 1859. This is a photograph by Franz Huber. This photograph was taken in the 1950s from Saudi Arabia. 420 million years ago, this organism existed. It was called Prototaxides. Prototaxides, laying down, was about 3 feet tall. The tallest plants on Earth at that time were less than 2 feet. Dr. Boyce at the University of Chicago published an article in the Journal of Geology this past year determining that Prototaxides was a giant fungus, a giant mushroom. Across the landscapes of Earth were dotted these giant mushrooms all across most land masses, and these existed for tens of millions of years. Now, we've had several extinction events, and as we march forward, 65 million years ago, most of you know about it, we had an asteroid impact. The Earth was struck by an asteroid, a huge amount of debris was jettisoned into the atmosphere, sunlight was cut off, and fungi inherited the Earth. Those organisms that paired with fungi were rewarded because fungi do not need light. More recently at Einstein University, they just determined that fungi use radiation as a source of energy, much like plants use light. So the prospect of fungi existing on other planets elsewhere, I think, is a foregone conclusion, at least in my own mind.
This is a fascinating talk, and I encourage you all to listen to the whole thing for it. But briefly, some of the things you tend to see if you look at this talk is, first, there's a lot of references to mycelia, which are kind of equivalent to roots. I guess it's the closest equivalent I could think of that everybody would know, right? We see the name Paul Stamets, which actually made it into the show. We see the reference specific to the genus Prototaxides, which you might remember is exactly the one they reference in the first season of Star Trek Discovery. There was a quote of organisms that paired with fungi were rewarded, which, as we saw, is exactly what happens with the tardigrade. And he has a very specific reference to these extraterrestrial fungi, which he sees as a foregone conclusion. I confess I'm a little bit more skeptical about that than he is, but nonetheless, the idea that fungal spores could have been introduced into space from Earth, that seems quite reasonable. Now, if you're thinking all this might still just be a coincidence, I want to show you one other thing. Here's a tweet from Brian Fuller, which you might remember is one of the people who started off the series, talking about this particular TED Talk, saying, watch this, 6 ways mushrooms can save the world. Clearly, this was the inspiration for the series. Now, could this lead to transportation across the universe? That's where there's, I think, some creative imagining there. My friend Dr. Erin McDonald actually has a great thing she presents in her talks about how maybe it's not actually the fungi themselves, but if you look at the fabric of the universe, it kind of looks like this mycelial network. Anyway, that's a little bit more figurative. I leave that to her, but nonetheless, if you think of it in a figurative way, there may be some basis to this. So, yay, science in season 1 of Star Trek Discovery. All right, let's move on to season 2 and also a little bit of connections to short treks. This time I'm just going to look at 2 broad categories of things rather than doing the little nuggets, since we already had our appetizers now. First thing, which I thought was really fun in season 2 of Star Trek Discovery, was a lot of discussion of Kelpian natural history. We loved seeing Saru in the series throughout, and it was nice getting a lot more backstory on what are the Kelpians? How do they differ from us? What's their natural history like? Now, they're obviously vertebrates. They seem to be mammals in the sense that you see the females seem to have what look like mammary glands, but let's dig a little bit deeper. First of all, they seem to have some senses that go beyond our senses. Very first one, of course, that we see right off the bat is the what I call the spidey sense for danger, kind of a Spider-Man reference there. And Saru has those little threat ganglia coming out the back of his neck, and when there's a threat, they kind of get erect or sticking out like that, right? Well, there are some animals that do this, that they like have little spiky things that become more erect when they're threatened. One you might think of is the African pygmy hedgehog. I don't know if any of you have ever had a pet hedgehog or knew somebody who had them, but again, when they're threatened, they erect their little spines. Now, that is not necessarily actually warning anybody or anything like that. It's actually to protect themselves. But something where in terms of warning, which might be what the function for these are, is vervet monkeys. They'll actually give this distinct alarm calls for different kinds of predators, like whether they're aerial predators or ground predators. So anyway, really cool. One of the things Saru seems to be able to do is he can smell emotion or anticipation, right? Now, we may not like to think about this, but we do exude emotion sense. And there was an interesting study done where sweat samples were taken from people who had watched a scary movie or not a scary movie, and then they were tested to see how their dogs reacted to smelling the sweat samples. Interestingly, the dogs definitely mirrored the owner's stress levels. They could smell this emotion from us. So again, this is based on some real science here. Last but not least, he has excellent vision. But in fact, not just excellent vision, but he has what they refer to as a wider optical window. What that means is he can see further into, say, ultraviolet light or infrared light than we can see. Now, kestrels, as an example, can actually see ultraviolet signals associated with rodent urine, and that's how they track down their prey. So again, there's a basis for this. Now, there's a big thing you might be thinking about in terms of the Kelpians. Now, this happened in season 2. And this is this big metamorphosis when the threat ganglia fall off and all of a sudden Saru's behavior changes. And they refer to this as the Vaharai. Unfortunately, they also refer to it incorrectly as an evolution. I want to stress, evolution does not happen in individuals. Evolution is something that happens across generations, not to an individual. So, for example, if you look at me in the 1970s versus me just a couple of years ago, I looked very different. I did not evolve, I just developed. Now, metamorphosis is a better word for this, and when we think of metamorphosis, we often think of like caterpillars changing into butterflies. There's a big change in an individual. Again, not an evolution though. Now, interestingly with this metamorphosis, not only is there a change in form, but there's also some change in behavior. You might remember Saru became a lot bolder, and there's a possible diet change, it seems like. So, for example, young Kelpians seem to eat this kelp-like stuff, okay? As they get older, they seem to like baul sandwiches. No, I'm just kidding. There seemed to be some sort of, we don't actually know if they were eating the baul, hopefully not. But nonetheless, there seemed to be behavior and possible diet change there. Now, there are frogs and toads that are herbivorous as tadpoles and carnivorous as adults. So again, mirroring this sort of kelp to baul transition. Again, we see this here in the real world, so yay, good science. Another sort of surprising thing we saw in season 2 of Discovery was these spikes. This is a great threat response where you could shoot the spikes at an enemy. When you think of that, when you think of an animal that can shoot spikes, what do you think of? You probably think of the porcupine. Unfortunately, you're thinking wrong. Porcupines actually cannot shoot their quills. They can kind of shake them off and have them kind of fly around a little bit, but it's not at all directed. Something that's a little bit better, but still not perfect because it doesn't really aim, is tarantulas. This is something actually I learned about at the Vegas convention when I presented a similar talk several years ago. They kind of do this. What they will do is they will essentially rub off their little spines, and those go flying about, and those can be damaging to a predator. That's pretty cool. But if you want something with actual aim, my favorite example is the bombardier beetle. This is a little beetle that, when threatened, will actually spray boiling acids, and it can actually aim it at a predator. That is pretty intense, but very good defense mechanism. Nonetheless, shooting something from your body is definitely something that we see here in animals on Earth. So again, really cool. And finally, last but not least, we see this change overall on the planet Kaminar, which is where the Kelpians and baul are from, in terms of the predator and prey dynamics. And this is something that obviously happened here on Earth over time. If you look, more than 65 million years ago, we had dinosaurs dominating the planet. There were some mammals around, but they tended to be small. They lived in terror of these dinosaurs. And then, 65 million years ago, we had this asteroid impact on Earth. There's a lot of volcanic activity, and that knocked back most of the dinosaurs. We still have the birds, which are basically the living dinosaurs. And now today, we see that lots and lots of large mammals here on the planet. Now, in terms of baul, maybe there was something else like that happened. Maybe since the Baul are a little bit more aquatic, we see them kind of rising up from the water, maybe they were less susceptible to some sort of natural disaster, and that's what triggered the turnaround where the Kelpians were no longer dominating the planet, but instead the Baul turned it over. We don't know. But anyway, it's a neat hypothesis, and again, it's a great connection to sort of real-world ecology over time. All right. Next big category I want to talk about from season 2 of Star Trek Discovery is lots of references to one of my favorite fields, genetics. Yay! All right, let me do a quick intro for genetics to make sure everybody's on the same page for some of the terms, and then I'll tell you some interesting examples from Discovery. First, DNA. Most of you are familiar with it. This is the hereditary material found in all life on Earth. Stretches of DNA with a message like turn hair color brown are called genes, and genes are assembled together onto chromosomes. All your chromosomes, all your DNA all together is referred to as your genome, and this is mostly found in the nucleus of your cells. Okay, I'll come back to that in just a second. Now, you get one copy of all your chromosomes from your mom, you get one copy of all your chromosomes from dad, and that's why you have 2 copies of all your chromosomes. And these affect how you look, how you feel, they affect how old you might grow up to be, things like that. They don't completely determine everything, but they have a big effect. Now, what you can do by studying DNA and studying genes and things like that, you can use these for identification, right? That we all have we have a unique genetic code that is unlike anybody else's, except maybe your identical twin. But this is something that's commonly used in forensics and other applications. And we saw this in the context of Captain Pike when he first joined the Enterprise, when he put his hand on there for a DNA match. We saw a reference to cloning. We have a mostly good but dark reference here where L'Rell is showing the severed head of Ash Tyler. Now, it turned out they said the severed head was a clone that was correct down to the genetic code. And it's true. If you were to have genetic code and somehow let something live that long, you would probably have a head that would look like the original head from Ash Tyler. Minus, of course, any scars or anything like that. So, yeah, it's a mostly good reference. There's a more mixed one in the context of Dr. Culber and his return. Apologies, this is a little bit of a spoiler. It was really good when Dr. Culber was reconstituted from his DNA in that he did not, in fact, have any scars, right? Because those are not encoded in your DNA. What wasn't so great was this reconstituted Culber somehow seemed to have all his memories. Now, those would not be in your DNA, so it's not really clear how that happened, but it's okay. There was a little bit of timey-wimey fun stuff in there. Now, I mentioned before talking about chromosomes, you get one copy from your mom and one copy from your dad for all the genes that are on chromosomes in your nucleus. Now, you also have genes somewhere else in your body. You have them in what is referred to as the mitochondria. The mitochondria, as you may remember from high school, is the powerhouse of the cell. Yay! I always love it when I say it at conventions. People very often remember that fact. What's interesting is the mitochondria also have their own little genome. It's much smaller than what you see in the nucleus. So for us, there's like 3.1 billion of those letters in the nucleus, times 2, because you have one copy from mom, one copy from dad. For your mitochondria, there's only like 16,000. Much, much smaller. Nonetheless, some of it is important. Now, mitochondrial DNA was important in Star Trek Discovery Season 2 because when they were studying this Red Angel suit, they said at first that it was associated with a genetic profile. And that's where they figured out, oh, this seems to be maybe associated with Michael Burnham's genetic profile. But they later specified it was in reference to Michael Burnham's mitochondrial DNA. Now, this is important because mitochondrial DNA is not inherited one copy from your mom, one copy from your dad, but in fact, your mitochondrial DNA is identical to your mom's. And Michael Burnham's thus was identical to her mom's, Dr. Burnham. So that was why either of them could fly this Red Angel suit, because they are genetically identical in terms of the mitochondrial DNA. That's awesome. Now, let me talk for just a second about how genes work in another reference that came up in the context of the Red Angel suit. So DNA is sort of the instruction manual of your cells, right? And I mentioned the genes or what have specific instructions for something. But proteins are the workhorses of the cell, right? So proteins are what make up your enzymes, your antibodies, they're more structural, things like that. So DNA encode what eventually become proteins, but they don't do it directly. There's an intermediary. So in between, your DNA will make what's referred to as messenger RNA, and that eventually, there's a couple of steps, leads to the proteins. Now, in Star Trek Discovery Season 2, there was a reference to Michael Burnham's mRNA being a perfect match to her mom's DNA. Now, I'm not quite sure why they did that, why they said mRNA, but it is true that any mRNA produced by the mitochondrial DNA of Michael Burnham would be a perfect match to the mRNA produced by her mom's mitochondrial DNA. So that's correct. I honestly wonder, though, if somebody thought M referred to mitochondrial, but nonetheless, it was accurate, even if it was a little bit funny. All right, let's move on to season 3. Now, season 3 is when Dr. Erin McDonald and I first became involved with Star Trek Discovery. She actually became involved with the whole franchise. I just did for Star Trek Discovery at this point. I had 2 contracts for season 3. One of them relates to prions, and the other one relates to an overarching plot of Su'Kal and the bird. I'm going to come back to the second one in just a second. On the first one, the contract I had with respect to prions was this ask of, okay, we have some aliens who are going to be arriving at the starbase. They're going to be sick. They're going to have a disease. And we need to know what this disease would be. And these are the parameters I was given. The disease has to be very hard to cure. It has to be fatal if uncured. It has to be associated with something they had eaten, specifically a fruit, because they were then going to have to go find the seed ship to understand it. And it has to be not communicable between people. They didn't want there to be some fear that everybody else was catching it. So my suggestion for this, and this is what actually got worked into the show, was that we use what are referred to as prions. Prion comes from the 2 words protein infection, and actually refers to an infectious kind of protein. Now, they're really interesting in that what happens is prions literally change other proteins to be like it, right? So this is going to be, let me show you what I mean by this. Now, when we think about proteins, we again tend to think about this sort of thread-like thing. And you produce proteins in your cells that have lots of functions, but these proteins have a 3-dimensional structure. They're not just this thread laying out like that, right? Now, there are some particular ones that actually are the ones associated with what we call prions that are produced in your cell at all times, right? I'm going to play a short video here. You can imagine these are being produced and they're going out to specific parts of your cell to do their function, and some are brought back and broken down. And you have this sort of turnover of these little proteins. And this is where it works well. Sometimes you have this other form that comes out and it changes other ones to be like it. It changes the 3D structure of other proteins around it to be like it. Now, what's bad about this, not just that it's changing them, but this new structure can't be broken down as easily by the cell. So what happens after it starts changing lots of these, you accumulate lots and lots of them because, again, they're changing all there's like it, more are being made, and they're changing to look like this prion protein, and they start accumulating. This is an actual picture of accumulation of prions across nerve cells in a human, or actually, I'm not sure if it's a human, across nerve cells, and they tend to accumulate again in nerves and in the brain. Now, there's a famous example of a prion disease you've probably heard of, not in humans, but in something else, and that's what's referred to as mad cow disease. You may remember this was a big problem, especially in Britain, but other places as well. Unfortunately, there's no cure. You may remember the way Britain dealt with this disease was basically sacrificing all the cows that had it. And even when we look at human diseases, I'm going to pull up, this is the website from the Center for Disease Control and Prevention here in the United States. It says, treatment of prion diseases remains supportive. No specific therapy has been shown to stop the progress of these diseases. This is bad. This is a bad disease. It's hard to cure and it comes from something eaten. However, for us at least, and for the prions we know, it tends to be associated with having eaten meat specifically, not from plants. So how do we adjust for this? Well, I did a little bit of literature searching in the scientific literature, and I found, in fact, there are some proteins in plants, Arabidopsis here is a plant, that actually exhibit a prion-like behavior. So it's not unprecedented that maybe something from a plant could do this. Now, what would have to happen, though, is there has to be a plant protein that's similar to a human neurological protein. And actually, I found one of those as well doing some more digging. There's a plant protein called RHD3, and that's very similar to a mammalian protein called atlastin-1. Atlastin-1, if broken, causes what's referred to as hereditary spastic paraplegia, which is a neurological disease. So, yes, there's some leaps here in terms of thinking that we're hoping that at some point in the future they find a plant protein that can exhibit a prion-like behavior, and that particular one can also affect a human or humanoid neurological protein. But it's possible. There's a little bit of leaps here, but the pieces are all there. And that's what we try to do with science consulting. It doesn't have to be perfect. At least has to just be the elements are there. Okay. Now, the second task was a little bit more challenging. This was one given both to Dr. Aaron McDonald and to myself. And we were told there was going to be an alien, a Kelpian, that was exposed to radiation. He was going to scream, and that all dilithium everywhere is going to go inert. And then we were told, science that. Felt a little bit like Sukhal here in the context of, oh my gosh, that's pretty hard. Nonetheless, my part to this was to look at the biology. And my part for the biology was we just need to make Sukhal different from other Kelpians. And importantly, we need to make him very radiation resistant. Because you might remember he was in that, I can't remember the name of the nebula. He was in a nebula there that had high levels of radiation. And that radiation was affecting Saru very quickly, but he was okay. Now, radiation, as I mentioned before, it can have negative effects on DNA. It can introduce breaks in the DNA. It can introduce mutations in the DNA. By mutations, I mean like changes in those DNA letters. Now, if you have two copies of your DNA, like most of us do, we have one copy from mom, one copy from dad, but you're having high levels of radiation there, you may not have a good template eventually. You may get enough stuff broken that you can't actually repair. It can't be repaired. Now, what I proposed is that maybe Sukhal, because of the radiation, was a polyploid. Meaning rather than having one copy from his mom, one copy from his dad, he may have actually had two copies from his mom and two copies from his dad. Now, radiation can potentially cause polyploidy to happen. But what's interesting about polyploidy is now that you have 4 copies instead of 2, even if some of them get damaged, you have more templates from which you can repair. And some microorganisms have more than 2 copies of their genome, and some scientists have proposed in scientific papers that this may make them have more likely templates for repair, and therefore better able to survive in radiation or other sorts of mutational conditions. Now, what this will also do, and when you're a polyploid, this also changes both the amount of protein that you're producing, as well as possibly some of the specific proteins that are being produced, right? So rather than being exactly like a Kelpian, he actually would develop a little bit differently, including potentially his vocal cords. So our proposal then is that maybe like an opera singer who can break glass, when he screams, he actually is screaming in some way affecting the dilithium. Now, specifically, one of the things we proposed is that dilithium, the di in dilithium, may refer to normal space and subspace components. And that when he screams, he's screaming at the resonant frequency of the connection between the normal space and subspace components of dilithium. And that's what breaks that connection and makes it so all of a sudden dilithium blows up. And since you're using subspace, it could happen everywhere faster than the speed of light. So, okay, there's a lot there. You probably want to read about this and think about this on your own. So I'll refer you to an article that Dr. Aaron McDonald and I wrote on StarTrek.com. It's called The Science Behind Discovery's Burn. But it's laid out in a lot more detail there. I just wanted to give you a snippet of what it was about. The punchline, again, is that we just wanted Sukhal to be different, such that he could survive the radiation, and such that when he screamed, he would actually break this dilithium connection. Presumably all the other Kelpians don't do that. Nonetheless, wonderful collaboration with Dr. Aaron McDonald, as always. Now, finally, I want to talk about Season 4 of Star Trek Discovery, and specifically this use of hydrocarbons for communication. Now, the task that was presented to me for this is that we were going to have a truly alien. And this is what's referred to as Species 10C. Now, with Species 10C, they wanted to have a new form of communication such that the universal translator was not good enough for translating it. And they wanted the translation to involve a process. It wasn't something that just, oh, here's the code, done, we figured it out. But there would have to be some back and forth and some thinking. So this was a really interesting problem. And I personally was really excited at the prospect of having truly alien aliens, because I would love to see more of that in Star Trek, honestly. But let's start at the very basics first. And we're talking about communication. What is communication? What are signals, right? Well, signals is basically the process of conveying information to another organism, and that message has been shaped by natural selection on the signaler, not necessarily the recipient. Now, why do I say that? Well, for example, we exhale carbon dioxide. That attracts mosquitoes. That is not us trying to signal to mosquitoes to come. That's actually bad for us. Now, what the steps involved in this have to be production, transmission, reception, and interpretation, right? Production, transmission, reception, and interpretation. So you have to produce the signal, it has to go someplace, has to be received, and then interpreted. Now, in terms of this, specifically looking at the transmission and reception pieces there, you have to think about what is the modality. Now, for us, the primary modalities that we use are visual or auditory, right? Now, there's one that we don't use so often, but a lot of other animals do, which is more chemical. Now, chemical for us would be based on smell or taste, things like that. We don't do so much communication that way, but this is something a lot of other animals do. Now, the most common term you've probably heard in this context is pheromones. Now, when people think pheromones, they always immediately jump to sex pheromones. That's not the only kind of pheromone out there. Pheromones are just chemical signals that have evolved for communication between members of the same species, right? Now, these are things that can either be broadcast in the air, or they can't be things that have to be touched directly. It's not just a scent, right? Now, one fun example of these is female moths, which release this sex pheromones, and males sometimes have these giant antennae, as you see in this picture here, for figuring out where's that female, and they can really zoom in on them. Now, we again are not great at chemical communication, but if you look, actually, there's some New World monkeys that actually have a whole other system that we don't have called the vomeronasal system. It's kind of like another smell system that they use for communication. We don't have it. A lot of Old World monkeys don't have it. We count as Old World monkeys in this context. So what I proposed for species 10C is that maybe they use what are referred to as hydrocarbons. These, as you would guess, involve carbons and hydrogens, right? Now, what's great about hydrocarbons is carbon, you may remember from its position on the periodic table, combined to up to 4 other elements. This allows for a lot more complexity than for some other things that don't have carbon in them. And these are good candidates because you can actually make all sorts of different and complex forms. So you see the pictures there on the bottom right. This is really important because if we're looking at an alien species and we're looking at things that are being elicited that affect us, it can't be super simple. So for example, it can't just be methane. It can't be something that we encounter all the time. Basically, people don't fall in love when they smell a fart. So this is a great opportunity to actually have some very complex things in here. And I was thinking of these hydrocarbons being kind of like word characters, right? And that was the way we framed this out. And I should stress, this was a wonderful collaboration where I was putting in some of the chemistry and biology, but there were 2 linguists who were involved in this as well, Douglas Vacach and Sherry Wells Jensen. Now, Sherry Wells Jensen is outstanding for this because she's also thinking about other modalities in that she is legally blind. They both work with METI. This is the Messaging Extraterrestrial Intelligence Organization. But this is a good demonstration that no one can do this kind of complex work alone, right? These are massive collaborations. We had lots of fun meetings with the VFX people in terms of what are the hydrocarbons going to look like, with each other about how does this translate into a language, and they proposed the Linkos language as parts of it. So lots of fun. And honestly, this sort of consulting mirrors what we do in scientific research, where everybody's bringing their own expertise and we're learning from each other. And amazingly, that's also what happened in the episode. You might remember they brought down Detmer and some other folks, and everybody was using their own expertise to help solve this problem of how do you figure out the communication. So what was happening in the writer's room matches what was happening on screen, matches what happened in real science. Lots of fun. Anyway, I don't want to go into too much depth on it because, it's really just this interesting idea of using hydrocarbons for communication, which various organisms on Earth do. Now, let me close out with a couple of words on science and science consulting. I told you a little bit about one of the things I love about this. But what I think is amazing is the networks actually pay for this. That is very impressive. They're actually spending money to bring people together with different backgrounds to make sure the science here isn't just crazy. Now, what they could do, and there are franchises out there, unlike Star Trek, which would just be like, let's have a freeze gun or a time stop gun or what, these sort of crazy things. But no, Star Trek knows about the number of people who are inspired to go into science, technology, engineering, and math because of representations on screen, and they want the science to be good. Now, there's a whole bunch of different scales at which consulting can happen. And I've done actually the first 3 of these. I'm not the last one. It could be a quick text check. It could be just here's a sentence. Can you tell me if this sentence is okay? Okay, here's the answer. And I actually have literally gotten some by text. There could be full episode help. So for example, the piece I mentioned about the prions was a little bit more of a part of an episode in terms of how does that work out. You could have a multi-episode or season arc help, which is what happened in the context of, say, the burn for season 3. Or at the most extreme, they could actually have a staff member on retainer, which right now the franchise has Dr. Aaron McDonald on retainer. Previously, you may remember Andre Bormanis was one of the Star Trek consultants. Again, it's wonderful they use people and expertise to make the science you see on screen at least reasonable. Obviously, there's creative license there. Obviously, plot will always trump science, but still, the fact they care about the science is great. And the other thing I love is that there's incredible enthusiasm for science. When you talk to the writers there, they're always like, oh, that's really interesting. Tell us more about it. And the one that always blows my mind is when they tell me, oh, what you do is so cool. I'm like, seriously? You make Star Trek. Like, what you do is cooler than anything. But there's a lot of enthusiasm for science and understanding the science and learning about the science. And it's heartwarming working with these brilliant people who are also just curious and excited and just incredibly kind. Now, as a consultant, this is a question that often comes up is, do you get to say no? Really, the best thing you can do is say yes and. Right? Essentially, because they're going to make whatever has to happen happen. Again, plot will always trump science. So if you can get them at least partway there, make it not crazy, things like that, that is better than just saying, no, you can't. That's not helpful to say no, you can't, because again, what they're trying to decide is going to happen has to happen for the plot. If you can't make it happen, sometimes it's better, just don't explain. And that's sometimes the answer. Sometimes people just say, all right, you know what, let's just have this there. Some future thing makes this work, and we're not going to talk about it. Great example, and this is a physics side, is the Heisenberg compensator, which makes the transporters work. Anyway, I should note just before I close out, a lot of the science you do, and this is true for, I'm sure, anything in show business, a lot of things that you do don't necessarily get used at the end. So there's some ideas that were floated, maybe some ideas that actually got fairly sketched out in detail, but maybe don't appear on screen, or the piece you were talking about doesn't actually show up. That's okay, right? It may be there, but not visible or not audible. Again, that's okay, because we're doing this because we love the project, not because we have to see this on screen. All right. I hope you enjoyed this talk. It's always a pleasure chatting with you. Feel free to follow me on socials or to subscribe to my YouTube channel, BioTrekkie Explains, and live long and prosper.